So in today's lesson, we will be looking at proteins. We're going to be starting off by looking at the structure of proteins. We're then going to move on to looking at peptide bonds, and then we're going to finish off by looking at the function of proteins. Okay, this is the part of the spec that we will be covering in today's lesson. It's basically everything about proteins, how proteins are made, the structure of proteins in depth, and basically sort of how proteins are made, condensation reactions, the bonds that are involved, and all the things like that. The only thing that we aren't going to be looking at is this point over here, because we've already looked at this in a previous lesson, in the biochemical test lesson. And this point over here, we will be covering throughout later sessions of the different proteins that there are available in different processes. So that point will already be covered elsewhere. Okay, so just an introduction. Now we've already looked at monomers and polymers, and this is building on from that idea. Now, amino acids are monomers from which proteins are made of. So if we were to draw a chain, like this one, this whole chain would be a polymer, and each repeating unit would be a monomer. Okay, now in proteins, this whole chain is the protein, that's the polymer, and the individual repeating unit is known as an amino acid. Okay? Each individual unit is known as an amino acid. Okay, and this is the general structure of a protein. Okay, now this is really important. I've seen this being asked in exams, so make sure you make a note of the structure. This is the structure of an amino acid. Okay, the key thing over here, you have the carbon in the middle. This is your central carbon. You have this group, which is known as the carboxyl group. And then you have this group, which is known as the amino group. This R we have seen in our last lesson already. This means variable group. So there could be anything on this R. It could be a CH2, CH3, H, anything. It can be anything. And then we have one hydrogen connected to the carbon. That's the general structure of an amino acid. Okay, that's what an amino acid looks like, and the structure. And basically, you need to know the structure of this amino acid because we're going to be looking at condensation reactions later on. Okay, so remember, we've looked at monomers and polymers. This is another example of monomers and polymers. Amino acid is the small repeating unit from which the larger polymer is made of. That's what I've represented here with these circles. And the circles are combined together. Many circles are combined together to form a longer chain, which is the protein or the polymer. And as you can see over here, there's a chemical bond. In the case of protein, this chemical bond is known as a peptide bond. But we will be looking at that in more depth. Okay? Now over here, the NH2 represents an amine group. So it could be an amino group or amine group, it doesn't really matter. The COOH represents the carboxyl group. So those are the two different groups that an amino acid has. And then the R group represents the side chain. So this could be variable group. It could be anything. So you could have, for example, CH NH2, and then you could have had H over there. If you didn't want H, you could have had CH2, or you could have had CH3. Yeah, it's a chain. It could be anything. Okay, and basically, you know, this is all represents variable group. There are 20 possible amino acids that are present in our body. Okay, so this R can be 20 different types of groups. Okay, there are 20 different variations of this R that there can be. In every amino acid, you will always have the amine group, the H, the carboxyl group, and the carbon. The only thing that's going to vary is this R group over here. And there are 20 variations that are possible. Okay, you don't need to know about the 20 variations. The spec just lists that there are 20 variations that there could be. Okay, so just to summarize the introduction of proteins. Proteins are basically long chains, as I represented over here. And because they are long chains, they are going to be made of subunits. And each of these subunits are called monomers, which we've already looked at in our first lesson. And when it comes to proteins, these small subunits are known as amino acids. Okay, they will combine together. Many amino acids will come together, they will combine, and then they will form a polymer. And the polymer is known as a protein. Now, how these amino acids combine together, you can see that there's a chemical bond being formed, just like we saw with lipids, we saw with starch, cellulose, and glycogen, carbohydrates, and monomers and polymers. The name of this chemical bond is known as a peptide bond. Okay, we also looked at the structure of an amino acid, which is over here. Each amino acid will always have a central carbon, an amine group or an amino group, a H, and a carboxyl group. The only thing that's going to change is this R group over here, and there are 20 possible variations of this R group. Okay, so that's basically the intro to amino acids. Remember, guys, if you have any questions, please do let me know at any point in the Q&A. Now, we've already looked at condensation reactions. Remember, condensation reactions is basically the formation of a chemical bond releasing a molecule of water. So forming a chemical bond and releasing a molecule of water. Okay, so we're forming a chemical bond and we're releasing a molecule of water. Okay, and we've already looked at some examples, starch, cellulose, glycogen in the past. Okay, and now what we're going to do is we're going to look at some examples with amino acids. So as you can see, these are two amino acids. Now, what can happen over here when these two amino acids line up together? Imagine, yeah, I was drawing circles and a long chain. Imagine that this was a big circle and this was a big circle. Okay, can you see that chemical bond is forming between the big circles? Now, if you go to that in a bit depth, okay, I'm just going to draw two amino acids out over here. Okay, so we've got C, R, H, N, H, H. Okay, that's one amino acid, and I'm just going to draw another one out. Okay, I've drawn out two amino acids. Now what I'm going to do is I'm going to change the color of my pen. So as you can see over here, what's going to happen is that, look, we have a OH over here and we have a H over here. Okay, and remember, whenever we're forming a chemical bond, we need to release a molecule of water. So what we're going to do is we're going to take this OH and H, and as you can see, two H's and one O means H2O. What we're going to have is that these two are going to be released. And now because these two are being released, the C is now no longer going to be bonded with the OH or the H. It's going to bond directly with the N. So what we're going to end up forming is this. So we're going to have N, C, E. this is what we're going to form. As you can see, because there was a H on one side and then there was a OH on one side, we've just combined them together to release a molecule of water. And because we've released the molecule of water, what we formed over here, I'm just going to switch pen again. What we have formed over here is this bond over here. And this bond is known as a peptide bond. Okay, and it's a condensation reaction because we've released a molecule of water in the process. Okay, now this structure now that we have formed, this structure over here, this is known as a dipeptide. Why is it known as a dipeptide? Well, simply you have two amino acids joined together. You had one over here, and then you had one over here. You had two amino acids, that's why we have formed a dipeptide. Okay, di means two, peptide is amino acids. Okay, dipeptide is the name of the structure which is formed because there have been two amino acids which have come together, they've combined together, they've formed a chemical bond in a condensation reaction. We call this structure dipeptide. Now, if this process kept occurring altogether, imagine what we just drew over here. Imagine we had another amino acid, yeah, and this was connected to another circle, another circle, and so on this side as well. Can you see we have many of these circles? Because we have many of these circles, what's going to happen is that it's going to change into a polymer. So this was a dipeptide. We only had two monomers together. Over here, we had many monomers together. Because there are lots of monomers together, it will change into a polymer. Okay, now proteins are a polymer, but there's a fancy word that we can use, and I've got that over here. Okay, so dipeptides are formed by the condensation reaction of two amino acids, which we've just seen over here. We had two amino acids, one over here, two over here, and when they came together, we saw that this molecule of water was being released and a peptide bond was being formed. Because a peptide bond was being formed, we formed from two separate amino acids, they became one, and that structure was called the dipeptide. Now, we saw that if this process continued to occur, you would form something like this. Okay, originally we just had this, okay, and this was called a dipeptide. Now, if this continues to occur like this and becomes a really long chain, this is now a polymer. If this is a dipeptide because there are two amino acids, and over here there are many amino acids, poly means many amino acids, this is going to become a polypeptide. Okay, so dipeptide, two amino acids. Polypeptide, poly means many. It's going to put times on you. Okay, and those are the two key words that you need to be familiar with. Okay, so a condensation reaction, we already know that a chemical bond is going to form and a molecule of water is going to be released in the process. Dipeptides are formed by the condensation reaction of two amino acids. Di means two. And then polypeptides are formed by the condensation reaction of many amino acids. Okay, just like we've looked there, we've got many amino acids, many circles joined together. And because we've got many circles joined together, that means it's a polypeptide. Poly meaning many. Okay, now that was basically the overall structure of a protein. So what we've just looked at is what the amino acids are made of. Okay, and what happens, how do amino acids combine together? We're now going to go into a bit more depth into sort of how proteins are constructed. So we know that proteins are a sequence of amino acids. We know that proteins are made of many amino acids. Okay, just like we looked at before, we saw proteins are basically this, where each individual circle is an amino acid. Okay, now you need to know there are four stages in a protein. You have the primary structure of a protein, you have the secondary structure of a protein, you have the tertiary structure of a protein, and then you finally have the quaternary structure of a protein. And for each of these structures, you need to know a definition, the bonds which are involved, and any additional key detail. And I've highlighted the parts that you need to know in yellow. Okay, so first of all, the primary structure of a protein. Now, the primary structure of a protein is basically the order or sequence of amino acids in the polypeptide chain. Okay, so for example, over here, this is a specific sequence of amino acids. Okay, now if I was to draw this a bit differently, which I am, just a minute. Okay, if I was to draw this a bit differently, okay, this is a specific sequence of amino acids, yeah? We've got a triangle, we've got a square, we've got a circle, we've got a triangle, we've got a square, and then we've got a circle. That's the first one. Now the second one is going to be circle, triangle, square, circle, triangle, square. Okay, now these are two different sequences of amino acids. Think about it. If you have a chain, if you had a chain, the building blocks are the ones which are the most important. Because these two are different chains, because they have a different sequence of amino acids, each of these shapes are representing an amino acid. Because each of these have a different sort of order of amino acids, this means it's going to fold up into something differently because there's a different sequence, of amino acids, it's going to fold up differently. Okay, so this is different. This first chain is different to this chain. Okay, and that's basically what the primary structure of a protein means. It's talking about the individual amino acids. So if I had different chains of amino acid, different sequences of amino acids, um, for example, if I had in one chain I had 3 amino acids and one chain I had 5 amino acids, those two chains are going to fold up into something completely different because there's a different number of amino acids in both of the chains. Okay, so what the chain consists of and the order of the amino acids is really important. And that's what basically determines the primary structure of a protein. Just like we had a look over here, these two chains are completely different because it's a different order of amino acids. And amino acids will build up together, and then proteins that are formed will go on for different functions. For example, enzymes, which are really important in the body. If an enzyme had a different shape, for example, you would have learned this at GCSE, it will no longer be able to bind to its substrate. It will bind to a different structure and catalyze a different reaction if it had a different active site or different sequence of amino acids. So that's why the primary structure is really important because it determines the sequence of amino acids. And because it determines the sequence of amino acids, it determines the overall structure of the protein. If the structure of the protein is different, that means, for example, in enzymes, it will no longer be able to catalyze the correct reaction. It will go on to either catalyze a different reaction or won't catalyze the reaction at all. Okay? A polypeptide, remember, a polypeptide is just another word for a sequence of amino acids or a protein. is made up of lots of amino acids joined together by several condensation reactions. So we've already looked at this on the previous slide. Basically this. Okay, many amino acids joined together in a chain through a series of many condensation reactions, just like we have there, releasing many molecules of water. Now, you also need to know the bond which is involved in the process. The bond which is involved is known as the peptide bond. Okay, we've already looked at peptide bonds. Remember, peptide bond is the formation of a chemical bond between the carboxyl group of one amino acid and the amino group of another amino acid. So in the primary structure, the only bond which is involved is the peptide bond, and the peptide bond is occurring between these amino acids. Okay, so we've got the peptide bond which is occurring between these amino acids. So what you need to know, the definition of the primary structure. Primary structure is just the sequence of amino acids in the polypeptide chain. And you need to say sequence, not just chain, because on a morph scheme, it will be in bold. You need to say the sequence of amino acids. Okay, and the bond which is involved is the peptide bond. You also need to state where it occurs. It occurs between the carboxyl group of one amino acid and the amino group of another. Okay, and that's basically the primary structure of a protein. Okay, now the secondary structure of a protein is basically, now we've got the primary structure. What do we have? Because we've got the primary structure, we have this structure. We have this sequence. We just have the sequence of amino acids. Now, remember, proteins can be used in many processes for a sort of, for example, enzymes. Now, if we just have a long chain like this, it's going to be pretty useless. Okay, it needs to be processed a bit more in order to be useful. And that's basically what happens. The polypeptide chain is processed. Okay, so you know this chain of amino acids that we have built up, it is processed. Okay, and it will process so that the chain can fold into a specific shape. So this is the highlighted part. It can either fold into an alpha helix shape or into a beta pleated sheet. So an alpha helix is something like this, something like that. It can fold into something like that, and a beta pleated sheet is something like that. Okay. So that's the key bit of detail that you need to know for the secondary structure. So we know for the primary structure, it's the sequence of amino acids and peptide bonds are involved because peptide bonds are present between the carboxyl group of one amino acid and the amino group of another. In the secondary structure of a protein, this primary sequence, the sequence of amino acids, is processed, and the long chain will fold up into an alpha helix or a beta pleated sheet. Okay, and again, you need to know the bonds which are present. Okay, hydrogen bonds are present this time. Okay, and if you do chemistry, you will know what hydrogen bonds are. Hydrogen bonds are basically hydrogen from one amino acid bonded to the oxygen from carboxyl of another amino acid. Or you could have the N from an amino group of another amino acid. That's basically what a hydrogen bond is. Okay, so in the secondary structure of a protein, what happens is that the chain is processed further and it causes the chain to bend into an alpha helix or fold into a beta pleated sheet. Hydrogen bonds are present in the secondary structure, and they are present between the carboxyl group, specifically the oxygen of one amino acid and the hydrogen of another amino acid. Okay, but it can also be present from the nitrogen of one amino group as well. Okay, now we've looked at the primary and secondary structure. The next structure is the tertiary structure of a protein. And guess what? Over here, so at the moment, we've got the sequence of amino acids and it's being processed. Well, over here, it's going to be folded even further because we want a unique structure. So it's just going to be folded even further and it's going to be folded into a unique 3D shape. Learn that phrase there, unique 3D shape. Okay, because obviously we know we don't want to deal with 2D here. Okay, because well, enzymes are there to catalyze reactions in our body. If they're 2D, they're pretty much useless. Okay, so we want to be dealing in 3D. So what's going to happen is that the secondary structure, where you have the alpha helix or beta pleated sheet, it's going to be folded up even further until you form a unique 3D shape. And this time, there are many bonds which are present. Ionic bonds, hydrogen bonds, and sometimes disulfide bonds. Disulfide, the clue is in the name, we've got sulfide. Disulfide bonds are present when you've got sulfur. Okay, they are present when you have sulfur. Okay, if you don't have sulfur, you're not going to have any disulfide bonds, but you will have ionic bonds and you have hydrogen bonds, both ionic and hydrogen. Okay, the ionic and disulfide bonds form between the R groups of different amino acids, whereas the disulfide bonds occur between sulfur. Sulfur has to be present for the bonds to occur. Okay, so S to S, that's a disulfide bridge or a disulfide bond. Okay, again, learn the highlighted information. Tertiary structure of a protein, the secondary structure is processed further until a unique 3D shape is formed, and then you will have ionic and hydrogen bonds present between the R groups of different amino acids and disulfide bonds if there is sulfur present. Okay, between two sulfurs. Now, finally, the quaternary structure of a protein. Not much to this. It's basically, and it's not really even part of some proteins. Quaternary structure is basically a protein that is made up of more than one polypeptide chain or more than one tertiary structure. For example, hemoglobin is made up of 4 polypeptide chains, and we're going to be looking at the structure of hemoglobin later on in a later lesson. Okay, so the quaternary structure is basically made up of more than one polypeptide chain. Okay, remember the tertiary structure, until we're making the tertiary structure, um, that's the polypeptide chain. So over here, until this point over here, when it's formed the unique 3D shape, that's one polypeptide chain. Quaternary structure forms when you have more than one polypeptide chain. So for example, you have this, you have another polypeptide chain linking in like this. That's 2 polypeptide chains coming together. So something like that. Quaternary structure has more than one polypeptide chain. And ideally, you just need to know that one bullet point there. And finally, guys, before we move on to some exam questions, what happens if a protein denatures? Now, you would have looked at this at GCSE, but if a protein is denatured, what happens is basically we've got bonds, right? We've got hydrogen bonds, we've got ionic bonds, we've got many bonds which are present within a protein. If the protein is denatured at high temperatures or low pHs, what actually happens is that the bonds which are holding the tertiary structure and secondary structure in place, they're going to break. They're going to break. And if they break, the 3D structure is lost. Okay, so for example, enzymes will no longer have the unique active site. Okay, so remember, we had hydrogen bonds and ionic bonds. which were present between the tertiary structure, and we had hydrogen bonds present between the secondary structure. If the protein denatures, for example, if the temperature is too high, or the pH is too high, or the pH is too low, and something happens to the bonds, well, because something has happened to the bonds, it's no longer able to sort of hold its shape. It's just going to break away, and the protein is going to lose its unique 3D structure. Imagine you're sticking something together with glue. You're sticking many bits and bobs um, with glue, and then the glue loses its efficiency because of something. For example, you pour water on it and it loses its efficiency. It becomes weaker. What's going to happen to the whole structure that you've just built? It's just going to collapse because the glue is now no is not strong enough to hold the structure in place. Okay, and that's basically the purpose of these bonds. These bonds are basically there to hold the structure in place. And if something happens to them through anything and they break, then the 3D shape is not going to be held together in its unique shape. So it's really important that we keep at our optimum temperature, not too high, not too low, and optimum keep pH as well.